go back to last week and cover the enzymes. That was where we left off. We stopped talking about prions, right? And that whole scary prospect. So I'm going to pick up where we left off, just a few slides on enzymes, which is kind of nice because it ties in a little bit into lipids. We'll talk about the different types of lipids. There's 6 main types of lipids, and we'll go into each of them and discuss them, and then talk about what kind of reactions these lipids undergo to either make them or hydrolyze them. But first, enzymes. So enzymes are proteins, and as you probably know, enzymes work by binding the transition state, and when they do, that ends up lowering the activation energy, right? So that hump that it has to get over goes way down, and as a result, the reaction happens much more quickly, okay? In fact, we talked about carbohydrates and polysaccharides. Let's say, for instance, glycogen. That's how we store our sugars, right? And then when we need those sugars, we break them off into monosaccharides that can then be used to undergo what's called glycolysis to then generate ATP. Okay, so sugar gets turned into what's called pyruvate. I don't know if we discussed that or not. Pyruvate then is converted into acetyl-CoA, which is a coenzyme, which we'll talk about in just a minute. And then that acetyl-CoA gets converted into carbon dioxide and water. And with that, there's a release of energy. And that energy release is what actually tacks on a phosphate group onto ADP to make ATP. Okay, so that's kind of the nutshell, right? But in order to break down polysaccharides, you need enzymes, right? Sugar, table sugar, that is a disaccharide. It is sucrose. It's made of glucose and fructose. And have you ever watched your table sugar just break down into monosaccharides? I mean, it's going to take a long time without enzymes to break stuff down. In fact, sugar, polysaccharides, glycogen are broken down by an enzyme called glycosidase, or glycosidase. It breaks down polysaccharides in the order of something like 10 to the 17th. Okay? Faster than without. Which means what would take sugar millions of years to break down is done in milliseconds, okay? By doing this process here. Without enzymes, we would not be alive because the functions that maintain homeostasis wouldn't happen in the quick amount of time that it needs to, right? Your cells responding to the environment requires signaling, signal transduction from one molecule to the next to then tell the nucleus to make a certain protein. We'll talk a little about that next week. But that's facilitated with enzymes. Otherwise, by the time your cell got that message, you'd be dead, okay? And so, the reason we see it like this is because there are different stages. We have, at first, the enzyme coming into contact with the substrate, okay? And that lowers the energy. And then we have this product, product's a bad word to use, but this next step where we have the enzyme and substrate bound together, okay? Then that forms the enzyme-bound product, what we're catalyzing, and then they separate out. Okay.
So they're like transition states, kind of? And the enzyme
So you
starts
can think
binding.
of different transition states for each of these steps. Enzyme and substrate come together, they bind, the enzyme does its thing, it releases. So kind of a 4-step process.
Okay.
And here are some examples of enzymes. You don't need to know these, because this is OChem, not biochem, but they do all kinds of different things. So, oxidoreductase, one example of that is dehydrogenase. Alcohol dehydrogenase is an enzyme that processes alcohol. If you were to consume that in your body, converts it into vinegar and formaldehyde, which is not a great alternative. We have transferases. So transferases are things like kinases, and a kinase is going to be that signal transduction that I talked about. So a molecule signaling to another molecule, signaling to another molecule, to another molecule, then goes into the nucleus. That signaling is done by transferring a phosphate group. So it's called phosphorylation. So one molecule, it's like, anybody watch, or, well, watching is probably, have you read Lord of the Rings trilogy, or seen the movies? Have you seen the movies? Well, great, this is not gonna work. But, okay, so anyway, it takes place in Middle Earth. They don't have cell phones, or satellite, or radio, or anything. And so, one kingdom is being attacked by the orcs, and they need backup. They need help from another kingdom. Okay, they need reinforcements. And they're wondering, they're sitting waiting, are they going to come? And they can't call them and say, yes, we're on our way. And there's this big mountain range in between. And so when they decide that they are going to help them out, what they do is they have this guy, and he's on top of one peak on this range, and there's this big wooden tower, and he lights it on fire, okay? And so far down the mountain range, there's someone else with a wooden tower, and they see it, right? And so that's the signal, right? They light their tower on fire, and then it's seen by someone else down the mountain range, and so finally that message gets to the kingdom that Rohan, that we're on our way. Okay, that to me is how our cells signal in a way too. Okay, it's not that the receptor on the membrane of the cell receives a signal and then it migrates to the nucleus. No, it receives some kind of stimulus, some kind of molecular cue, which then leads to a transfer of a phosphate group from that receptor to a molecule that is in the cytoplasm. It gets phosphorylated. It then moves that phosphate group via an enzyme, these kinases, to the next molecule, and it just sequentially does that until it activates a transcription factor that then goes in and says, hey, make this mRNA so that it can be made into a protein so that we can respond to whatever is going on outside the cell. Okay.
So it doesn't like really know where it's going. It's just like a domino effect, and eventually it reaches like the target.
Yeah, right. So if we were to look up signal transduction, it's all different types, and this is often associated with cancer. What's a good one? We have these protein receptors here, okay? And G protein coupled receptors, integrins that integrate through the membrane. And what happens is some kind of signal here will bind to these receptors, and then that leads to this activation activating this molecule, which then talks to this molecule. This is a cell signaling pathway that leads to cell division. So when these RAS, RAF, and MEK, and MAPK, when those get mutated, one of these, then you get the signal. Let's divide. Let's just divide, okay? And so that's what cancer is, right? Unchecked cellular division. But it is a series, and so what a lot of cancer therapies will do is, okay, it will bind one of these molecules. So there'll be a RAS drug, okay? So it binds RAS so that RAS can no longer signal to RAF, and then RAF can't signal to MEK, and MEK can't signal to MAPK, and then MAPK can't go in and upregulate certain genes to lead to cellular division. Okay, but what happens, okay, well, cancer cells, because they're dividing so quickly, mutations happen randomly, and you get adaptations to the drug, right? And so, okay, well, if we can't do it this way, we'll activate it here, okay? So we'll just start going here, and we'll activate RAF via this PKC, right? But yes, it is. It's kind of like passing the torch from one to the other. This GRB2 doesn't go straight to the nucleus. It's a stepwise process, and that's important because if one of these things gets mutated, sometimes these can compensate, right? You want it to be stepwise so you can turn it on and off pretty quickly. As soon as you make a protein to some external stimulus, you need to stop making that protein, right? You don't want to just keep churning out the protein, right? So that's why it's very nice. It's very nice to be able to just use a phosphate group. Tag it, as soon as the phosphate comes off, the message stops. Right? And as we'll talk about next week, messenger RNA itself is very unstable. It degrades rapidly. So as soon as this gene regulation happens, it makes an mRNA transcript that comes out here, starts getting translated to protein, but then it degrades. Okay. If it's stuck around in the cytoplasm, the ribosomes will just keep making protein, and this whole cell will get gummed up with that particular protein. Right? DNA is very stable. We could swab these tables and get DNA. You're not going to get RNA off of these tables. As soon as the RNA comes out, and we'll talk about how that's remarkable because there's very subtle differences. There's not much of a difference between RNA and DNA. Structurally, I should say. All right, so lots of different enzymes. Lipases, that's going to be what hydrolyzes fats. So today we're talking about lipids. One type of lipid we'll talk about is a triglyceride, or what's called a triacylglycerol. That's the kind of fat that we store in our body. Okay, it's not the kind of fat that makes our hormones. It's not the kind of fat that, like cholesterol, that is a structural lipid. It's the kind of fat that we can use to make ATP if we don't have sugar. But it's, as I'll talk about, it is fats are non-polar. That's why oil and water don't mix, right? So if you consume fats, how do you get it in throughout your body when our bodies are mostly polar, right? You gotta break it down. So we literally take that triglyceride, break it up into fatty acids, let those kind of go through and be absorbed, or we have things like lipoproteins that can then bind them and take them across. And then once it's absorbed into our small intestine and into our bloodstream, then we reassemble them back into the triglyceride. And then we can either break it down again to make ATP essentially, or squirt it into our adipocytes. So anyway, lots of different things. Many enzymes will have what are called cofactors. Okay? And you can buy a lot of supplements that are cofactors or coenzymes. If you go to the supplement aisle, you'll see a lot of things that are called coenzymes. And so the idea behind that is that these are not necessarily proteins. They're not enzymes, but they are required to help facilitate enzymatic activity. Okay? And so they can be things like ions. You need to have magnesium. A lot of human beings are deficient in magnesium. And that's a problem because you need magnesium for DNA synthesis. In fact, when we did PCR, you just had the master mix and you added primers, but you know, the way I would do it in the lab, the way mostly it's done is that you buy these reagents all separate. So you have the Taq polymerase in a tube, and then you have magnesium chloride in a tube, and then you have the what are called the DNTPs, or the nucleotides that are going to be tacked on in a different tube, and you mix them all together. The magnesium chloride is going to be a cofactor that helps with the addition, with the polymerase, helps it do its job. Okay. And then coenzymes, these are also going to be not enzymes themselves, but organic molecules that help reactions occur. So ATP, right? Adenosine triphosphate, that's the power molecule. It's not an enzyme, and it's not a protein, even. It's just an adenosine with 3 phosphate groups attached to it. But that transfers that phosphate to other molecules, and in doing so, allows them to do their function, whether they're enzymes or other things. We'll talk about acetyl-CoA. I think that might be the next slide. Is it? Yeah. So acetyl-CoA is important for what's called the citric acid cycle. Okay? The citric acid cycle is this long process where we essentially take acetyl-CoA and we turn it into a thing that can make ATP. All right. So you need some energy. You go to your sugar store, you go to your glycogen, or maybe you just drink or ate some sugar. That sugar, like I said, undergoes glycolysis, pyruvic acid or pyruvate, and then essentially gets converted to acetyl-CoA. Acetyl-CoA is just this acetyl group, and then coenzyme A. So it's I keep pointing here like it's different. I need to be able to have like previous slides, but yeah, it's a coenzyme. It's not an enzyme itself, but it facilitates that process. This is going to break down, as I mentioned, into carbon dioxide and water, and that energy release is going to tack on that phosphate onto the ATP. Okay. But in order for this to be turned into citrate, which is the first step in that citric acid cycle, you need to have oxaloacetate. Okay? So these come together, right? You can see that this is similar to this, right? Without this acetyl-CoA, this can't turn into our citrate that we need for that citric acid cycle step. Okay? And what you also need is this citrate synthase. So ACE enzyme, right? What enzymes do is they speed up reactions, like I mentioned, right? And one of the ways in which they do that is to bring these things together. Right? None of these things have wings or legs or flagella, right? So not only do they need to come together, but they need to come together in the right orientation, because it's like a lock and key, right? And so what these enzymes do is, they're proteins, right? And so they'll open up a cleft on one side to bind, let's say, the oxaloacetate. And then once they do that, they'll open up another cleft on some side and bind acetyl-CoA. And then, like, you know, they're the matchmaker. They bring them together in the right position in space to then allow them to make this, okay? But if you get rid of this, this is going to take a lot longer to happen, right? And we don't have that kind of time. Ain't got no time for that. Right? So that's the power of enzymes. Questions on that? All right.
So the enzymes just get used over and over again. They don't really get used up.
Yeah. They can be used multiple times. Of course, proteins have a shelf life, just like all biomolecules. So typically, it's not a one and done thing. They are a catalyst, and by definition, a catalyst is something that facilitates a reaction without being consumed itself. And so, yeah, enzymes make citrate. Oh, here, put you guys together, make some more citrate, release citrate, and then eventually, of course, that structure will start to degrade. And then you just have to make some more.
When it starts degrading, does it get tagged to get like broken down?
Yeah, so it will start to denature.
Yeah.
And just from it will denature, and then it will get recycled to a certain extent. So
cool.
We need to have proteins because we need to consume proteins because a lot of our proteins eventually leave us. But that said, we can recycle these proteins as well. So enzymes can be chopped up into their amino acids again, and then used to make some different protein.
Cool.
Okay, so these are the 6 main classes of lipids. They're broken up into 2 types. We have complex, we have simple. Complex means they can be hydrolyzed, or they can be broken up into simpler components, particularly fatty acids. Okay, so waxes are essentially made by joining together alcohol and carboxylic acid together. Triglycerides are going to be using a glycerol molecule, so an alcohol attaching 3 fatty acids to that alcohol. And then a phospholipid is going to be very similar to a triglyceride. The only difference is that instead of having 3 fatty acids, one of the fatty acids is removed and a phosphate group is put on there. So it just has 2 long fatty acids are long hydrocarbons.
The chains. Yeah.
So a phospholipid is so waxes, earwax, skin, lots of fruits and things have wax coverings. So wax is usually a protective lipid. Triglycerides are predominantly an energy lipid. Phospholipids are going to be what make up our cell membranes. You've all heard about the phospholipid bilayer, right? So that's the main functions of these complex ones. Simple lipids are ones that can't be broken down into simpler components. And these are going to be things like steroids. Some steroids are proteins, but some steroids are lipids. Cholesterol. Cholesterol is an alcohol because it ends in "-ol", right? But it's also a steroid, and it's a lipid. Prostaglandins, those are a type of lipid. I don't know, actually, I would label this icosanoid, which I think I got a slide that talks about. Prostaglandin is a type of icosanoid, and these involve things like you've probably heard of maybe lycopenes, potentially. They're in fruit and tomatoes, they give it a red color. Lycopenes are antioxidants, so some dietitians will tout eating tomatoes. the red color comes from lycopenes. Lycopenes are antioxidants, and so they've been associated particularly to be also protective of prostate cancer. So prostaglandin, prostate gland, that's where it gets its name. And then terpenes. Terpenes are predominantly in essential oils. Okay, so fragrances, flavorings, things of that nature are going to be terpenes. And so when you did your, you guys did extraction of essential oils,
Yeah, I
right?
think in the winter. Yeah.
Did you do like limonene and?
We did, yeah, like
Did you do
clove
lemons?
and lemons, yeah. Uh-huh.
So limonene
Yeah.
gets its name because it's a terpene. So that stuff, that oil is a lipid, right? Now, if it's vegetable oil, it's a triglyceride, but if it's an essential oil, it's typically going to be a terpene.
So what's like the common attribute that would like unite all these lipids? It's just like having like a lot of non-polar areas, or?
That's a great question. Yeah, so we'll go through all of these, and you won't really see a smoking gun for what makes these lipids.
It's a lot of different
In fact,
things.
some of these are more, they're not necessarily lipids, they're more lipophilic. So, it's the non-polar attribute of them that puts them under that category. Because they are structurally, they're going to be very, like triglycerides and phospholipids are similar, right? But waxes, there's a lot of them have esters. Okay, so some functional groups will be, you'll see in a lot of these, but not necessarily all of them. Okay, in fact, terpenes don't have really any functional groups, or don't have to have any functional groups except for double bonds. Okay, so nothing that really rings true,
Yeah.
other than you hit the nail on the head, they're non-polar.
Non-polar.
And so, again, with biomolecules, we think of them as, biology is polar, but we have non-polar attributes. All right, so these are the complex ones, and really what these all have in common are these long hydrocarbons,
That's crazy.
right? And so, as you all know, hydrocarbons are non-polar, right? And so it makes it easy to extract these out with organic solvents. Right? And here are some samples. So phospholipid lecithin, that's going to make up cell membranes, right? Here's a wax from the sperm whale, and triglycerides. There's all different types of triglycerides. So what makes one triglyceride different from another is whatever type of fatty acid it has, or I should say combination, right? It has 3 fatty acids, and there are many different fatty acids, and they differ based on how long their hydrocarbon chain is, whether or not they're saturated, whether or not they're unsaturated, and how many double bonds are in that unsaturated hydrocarbon. Okay, and so the permutation is not endless, but you can imagine there's going to be a lot of different triglycerides based on which 3 you pair together. Okay.
So the triglycerides and phospholipids are sort of like interchangeable. They can be converted. You can like pop off one of those chains. And
Yeah, in theory, you could just take off a fatty acid and stick on a phosphate group and make it a phospholipid. that being said, you have to stop it at just one. So lipases tend to just chop all three off.
Okay.
As I mentioned, when you consume fat in order to get it absorbed, the lipase is going to chop it off, and you end up putting hydrogens here, and so you make that's glycerol.
Okay.
Okay, and then these come off, and essentially are these fatty acids, and then once it's absorbed, you have enzymes that put them back together with that glycerol, because glycerol can pass through, be absorbed in the microvilli of the intestine. And then some fatty acids, if they're small enough, they can squeeze through.
Okay.
And if not, then we have these things called lipoproteins, which form kind of what a micelle is? A micelle is essentially like a soap bubble.
Oh, okay.
But
Great.
you have a polar head and a non-polar tail, and the polar head kind of forms this sphere on the outside to embrace the water, and then the non-polar tails are all on the inside, so you have this a globe that is a transportation vessel, and so lipoprotein, it's a combination of lipids and proteins, and that's going to be you've heard of good cholesterol and bad cholesterol. LDL high, that's LDL is low lipoprotein, and then HDL is high lipoprotein, high density, and that's so yeah, HDL, high density lipoprotein. Fat, lipids, and proteins have different densities, right? And so the ratio of lipids to proteins are going to make one type of lipoprotein more dense than another. So it's just an abstract way of classifying lipoproteins. High density lipoprotein is going to have less lipids on it, so it's going to be more dense. And so that is bad cholesterol, because what that does is that takes lipids and puts it in your blood. Okay? And that's how your arteries get clogged with cholesterol and stuff, because they start breaking down into the simple lipids. LDL, or do I have that?
You have this
I have it backwards, yeah. LDL is the bad cholesterol. That takes it to your blood. High density removes it from your blood and takes it and stores it in your adipocytes.
Okay.
So it's a one-way transportation. Okay. And I got sidetracked because I like biochemistry better. If you haven't put that together, but yes, you could trim off one. That's where you just got to be careful that you do it in that controlled setting. But that is the major difference. Okay, so here's some examples. Oh, here, right, limonene. That's what you got from the orange peel, right? Prostaglandins and cholesterol. And so as you can see, like, none of these there's no common denominator here in these simple lipids, right? A steroid, though, it's a sterol. The common denominator is they're all going to have these 4 cyclic rings, okay? They may have different substituents on there, okay? But they're all going to have this commonality here. There's not that present with the eicosanoids, but the eicosanoids are going to have a commonality in that it's going to have this this looping-like structure to it. Okay, so it's going to go one way, and then it's going to bend and come the other way. And maybe there's this cyclopentic ring, or maybe it's just not a cyclic ring. It doesn't necessarily have to have that on there, although a lot of them do have this, but it does have this folding around feature. And then terpenes, we'll get into what makes terpenes terpenes. things, but essentially they are going to have 5 carbons. One, or let's do it this way, with a branch. 1, 2, 3, 4, 5. 1, that was my elbow. 2, 3, 4, 5. Multiples of 5. And on the second carbon, there's a branch that comes off. Did you hear my elbow pop?
No.
No? Okay, good. Because it's starting to get louder and louder. It's making me feel older and older as I just move. I get these creaks.
Yeah.
It's like, okay. Questions so far on any of this? All right. Well, let's get into waxes then. Waxes are essentially high molecular weight esters, right? This right here, that is an ester. And they differ only in how long their side chains are, okay? So this one is predominantly what is used to make beeswax, okay? We put together a 30 carbon, this is probably the carboxylic acid. I guess it's depicting this probably as the alcohol. But anyway, we put an alcohol together and we put a carboxylic acid together to form an ester. And 16 carbons and 30 carbons makes it this wax. But other waxes could have shorter ones, right? So in the synthesis of carnauba wax, here we have a 30 carbon alcohol, right? And a 26 carbon carboxylic acid, right? So they all have this really long molecular weight. They have the same components coming together. So we bring together an alcohol and a carboxylic acid to make an ester. What differs is just how long those hydrocarbons are. And as you could imagine, wax has a pretty high melting point. Wax is a pretty dense substance. And that's because it's just hydrocarbons. There's no pi bonds in there. When we talk about triglycerides, pi bonds create kinks, okay? And so things can't stack on top of each other as readily. Here, these are all sigma bonds, right? And so you can imagine if we had another beeswax molecule, it would just fit really nicely underneath there, right? And then another one, we could stack them all, and what's going to keep them together? London dispersion forces, which is not a strong intermolecular force, but since these hydrocarbons are so long, there's a bunch, and then they're all in proximity to each other, there's no gaps, there's that London dispersion dispersed across the molecule, right? Making this such that, it has a high melting point. Of course, paraffins and stuff like that, or soy candles, that's a wax. Like those are all waxes, and they will melt, but fire is pretty hot, right? So it's not going to necessarily just melt in your hands. Okay. So now we're going to get into the complex lipids, and let's start with triglycerides. So a triglyceride is going to be what's used predominantly for energy storage. Okay. Triglycerides can release about 9 kilocalories per gram, which is more than sugar and protein. Proteins and sugars release about 4 kilocalories per gram. So you get more bang for your buck. And it turns out that triglycerides can make more ATP. Okay? So pound for pound, I guess I should say gram for gram. In a chemistry class, we should use imperial system. Gram for gram, you get about 3 times as much ATP. If you take these triglycerides and break them down to fatty acids, essentially what happens from there is it starts cutting these fatty acids further into little pieces that then become acetyl-CoA. And that's that coenzyme that goes into the citric acid cycle. All right? So these molecules can make about 3 times as much ATP than glucose. So why don't we use fat over glucose, right? It's got more energy released. It can make more ATP. Why is this the stuff we store? Well, because sugar, for one, is polar. Lipids are non-polar. So you can't mobilize this stuff as quickly. So right now, if you were to go running, your body would need to make ATP, right? It would say, okay, whatever sugar you ingested that day or that morning is going to be in your blood unless your glucagon has taken it to be turned into glycogen. You'll start using that because it's already in your bloodstream. It can live in your bloodstream. Now, you don't want too much to. We talked about carbohydrates. We talked a little bit about diabetes, right? But then you start burning that up, and your muscles need more ATP. So the liver will start breaking down the glycogen stores, and it can do that quickly. Get it to your blood, send it to your muscles. Right? Once you've burned all of your sugar, though, and your glycogen stores, you still need to make ATP. And you can either go and do that and have anaerobic respiration, which will happen. That'll lead to lactic acid production and build up in your muscles. That's why you get sore. But it will also start going to your adipocytes and say, okay, let's pull these triglycerides out and then start cutting them up. Okay, but that, like I said, is inefficient. That takes time. So even though it's a lot more energy, it's not as readily available for immediate use. And so people who do long distance things, it's actually from like, I'm sure there's all different types of, nutrition guidelines and dietary, tips, but from a biochemistry perspective, the best thing is to have some carbohydrates the night before so that you can build up your glycogen stores. Don't have a lot of sugar beforehand, so the carb loading thing should happen the day before, not right before. Anybody's watched The Office? I don't remember if you guys watched The Office. Did we talk about Michael Scott and his race to cure rabies?
With the pasta?
Yeah, he eats a bunch of pasta Alfredo from styrofoam. He has Dwight go and get him something. You don't want to do that. He ends up throwing up, right? It's Alfredo sauce, which is not going to sell on a run. But eating a bunch of sugar right beforehand is going to start modulating your insulin and glycogen signaling. Okay, and so that's going to start moving that sugar around before you necessarily want it to. So you carb load the day before, build up those stores, and then right as you're starting to do your event, then you might do like a shot of glucose or a shot of some kind of carbs, and then if you don't have any more sugar in your system, you're gonna hit bonk, you're gonna hit the wall, right? Your muscles are gonna feel that fatigue, and that's because it's saying, okay, we gotta go to the fat. Well, it's taking a while. What are we gonna do in the meantime? We're gonna do anaerobic respiration. That's building up lactic acid. It's not as efficient. I'm not getting enough ATP to my muscles, and so it feels like I've got concrete boots on, all that stuff, okay? But a triglyceride is essentially just a glycerol. So a glycerol, you have alcohols, you have diols, and then you have glycerols, 3 alcohol groups. And so each of these end up through a dehydration step tacked on these fatty acids. What's a fatty acid? Fatty acid is a carboxylic acid with a long hydrocarbon tail. It could be like I said, it could be saturated, it could be unsaturated, it could be monounsaturated, it could be polyunsaturated. But through that dehydration reaction, we end up making an ester here. Okay. And that is what's then going to break down. Again, to be absorbed, it either goes into those lipoproteins, or we have lipase break it down into glycerol and these 3 fatty acids. Get absorbed and then they come back. So this is a reversible process, right? Here are different fatty acids: lauric acid, myristic acid, palmitic acid, stearic acid, etc., etc. And you don't need to know these, but what I do want you to appreciate is the melting points, okay? 2 things determine the melting points of these fatty acids. One is the length of the hydrocarbon tail. Right? The longer the tail, the more London dispersion forces, the higher the melting point will be. The other factor is going to be saturation. If it is unsaturated, it's going to cause these kinks here at that double bond. The kink means now that they can't lay nicely on top of each other, and so it's going to be more like this. Less London dispersion forces to keep them together. So not only is it the number of not only is it the presence of double bonds, but the number of double bonds. They can be cis or trans. Okay? So if we look here, here are saturated, here are unsaturated. Here's palmitic acid. It has 16 carbons. Its melting point is 63. Right? Palmitoleic acid, also 16 carbons, but it has 1 double bond. Just 1. So that's mono unsaturated, and its melting point is 0. Okay? Big difference. This is Celsius, 63 degrees. That's a lot hot. That's a lot hot. It's a lot hotter than freezing. Okay? And that's why vegetable oils are liquid at room temperature, because they are unsaturated. They are unsaturated, so they have double bonds. So think about your different oils that you might use. Avocado oil, right? Olive oil, canola oil, they're all liquids. Now, coconut oil, that is more of a solid, but it's mono-saturated. So that means it just has 1 double bond. Olive oil, avocado oil, those are all polyunsaturated. Okay? But if you think about it, that coconut oil melts really quickly, right? So it is a solid at room temperature, but if you heat it up just a little bit, it'll melt. Butter comes from animals, right? Animal fat is predominant. plant fat is predominantly unsaturated. Okay, so think about butter, it's a solid at room temperature, right? Anybody cook bacon? Anybody let the grease cool down at room temperature? It's a solid. Anybody cook with that grease? Yeah, you know,
Not
right?
regular, but it's rich.
Saturated fats are not advised for high consumption, right? For a cardiovascular perspective. There are other philosophies that say, yeah, but saturated fats don't get oxidized. And we'll talk about, when we get to reactions, we'll talk about how unsaturated fats can be oxidized. And that's in fact why certain oils have a cooking temperature threshold on them. So if you look at a bottle, it'll say, and you probably know all about
The
this.
smoke point.
Yeah, the smoke point. Or certain oils will get rancid after a while because they get oxidized. And so there's a whole school of thought thinking that you shouldn't have unsaturated fats because they get oxidized in the body and can lead to free radicals and stuff like that. I'm not going to advise, like I said, I'm not a nutritionist, so I'm not going to advise either one, but I tend to think there's a lot more data showing that we should limit saturated fats over unsaturated fats.
What causes like some oils to have a higher smoke point? Is that related to the like saturation or?
Yeah,
Uh-huh.
precisely.
Okay.
The degree of pi bonds
Mhm.
is in each fatty acid, or how many, like, so you might, so it's 3 fatty acids, right? It's a permutation. You could have 2 saturated fatty acids and then 1 unsaturated.
Okay.
Okay, that's going to be different than something that has only saturated versus something that has only unsaturated, right? You can have different permutations of these, and so depending on, like, this is an icosanoid. You see how it has that wraparound? It doesn't have the cyclopentic ring at the end. This is arachidonic acid. We'll talk about that when we get there later, but that's what is used to make prostaglandin and all these other things. It's also what leads to inflammation. But different fatty acids have different numbers of degrees of unsaturation. So yeah, different smoke points, different shelf life is going to be based on which one's the range of.
Like how easy they get oxidized.
Yeah. And in fact, the FDA, has guidelines on how long certain oils should be left before they are consumed, because if they get oxidized again, that creates free radicals and that can be toxic. So you don't want to use old vegetable oil. Not vegetable oil is a mixture thing, like that's its own thing. Like plant-based oils, you don't want to use them if they've been laying around for a long time. In addition to making your food potentially taste bad,
Yeah.
it will be bad for you as well. All right. Okay, let's do this, and then we'll take a break. Which triglyceride, based on those 2 criteria that I mentioned, right? The length of the hydrocarbon tail and the number of pi bonds, which one is going to have the higher melting point?
Called 19.
Yeah, the unsaturated one.
Yeah.
What's that?
Probably the unsaturated.
Yeah.
Or like, no. Yeah, the saturated. That's what I meant.
So yeah, you gotta think about it. So if you
yeah.
have a higher melting point,
So
then
you
you're
should
going to be a
focus on
solid
the
at a higher temperature. You're going to be a liquid at a lower temperature. So when you see, let's use the first criteria. Okay, how many carbons? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Same. It looks like this is one more, and that's one more. So hydrocarbon numbers are all the same. So that's not going to differentiate these. If these were all saturated, then they would have probably about the same melting point, right? But because this one has unsaturation, right, has these pi bonds, this is going to have a lower melting point. They're asking for the higher one, it's going to be this one. And I also want you to pay attention to the fact that, what is this? Cis or trans? Cis. Most of these are going to have a cis conformation. You've heard of trans fats, probably? Yeah. There's a reason trans fats have been phased out. All right, perfect. Let's take a 10-minute break.